And I guess we'll move on to Aude next. And yeah, if you want to continue to put your questions into the chat box for Aude during her presentation, feel free to do so. And we'll run through them at the end of her presentation. Thanks again, Paris. All right, Aude.
Yes.
The microphone is yours.
Thank you. Can you see it well? Perfect. Great. So hi everyone. I'm really honored to have been invited to present this data. So I did this project in Severin Bois's lab when I was in PhD at the Price Brain Institute, and it was about the implication of peripheral macrophages in amyotrophic lateral sclerosis. So amyotrophic lateral sclerosis, or ALS, is the most common adult onset motor neuron disease, and it's characterized by a progressive muscle atrophy and a progressive paralysis until the ultimate death of the patients within 2 to 5 years by respiratory failure. The mean disease onset is between 55 to 60 years old, even though there are also younger cases that exist. So most of the cases are actually sporadic without any familial history, but 10% are familial cases, and those cases allow scientists to discover a lot of genes that are implicated in the disease. The 4 major ones being C9orf72, SOD1, TARDBP, and FUS-TLS. So we are going to focus today on the gene SOD1, who has been the first gene discovered linked to ALS back in 1993. It encodes for the superoxide dismutase 1, which is a detoxifying enzyme, and the mutation in the gene leads to ALS through an unknown gain of the toxic function of the protein. And so very shortly after its discovery, there are mouse models that have been created to study the disease, and they are still the most used mouse models to study ALS so far. So when the mouse overexpress the human wild-type form of the SOD1, they do not display an ALS-like phenotype. But when they overexpress the mutant form of the human SOD1, they have a progressive motor neuron degeneration and inflammation, and they actually have an ALS-like phenotype. So I'm not going to go into details, but know that there are both cell autonomous mechanisms and non-cell autonomous mechanisms that are contributing to the disease. And so today we are going to talk in particular about the peripheral macrophages and a little bit about the microglial cells that are actually involved in the disease. So microglial cells have been shown to microglial cells have been shown to be activated both in ALS patients and in ALS mouse spinal cords. And in 2006, 2 different papers have actually shown that when we reduce the mutant SOD1 in the microglia and the peripheral macrophages altogether, that leads to an increased survival of the ALS mice. However, those studies couldn't discriminate the role of the microglial cells and the role of the peripheral macrophages. And so we were interested in the peripheral macrophages because the motor neurons has its axons like laying at the periphery and extending to the muscle. And so it's surrounded by the peripheral macrophages, while its cell body is surrounded by microglial cells. And it has been shown both in human and mice that the axon degeneration is actually a primary event in the motor neuron degeneration. As I said, both microglial cells and peripheral macrophages are involved in disease progression. And also both populations are macrophage type cells, so they share common features. They have different developmental origins, and they are also evolving in different environments, and that could lead to different answers to the motor neuron degeneration. And finally, of course, by being at the periphery, they are way easier to target than the microglial cells in the central nervous system. So we wanted to know what is the implication of peripheral macrophages in ALS independently of the microglial cells. So for the project, we used 2 different mouse models of ALS, the SOD1G93A mice that have a fast disease, and the SOD1G37R mice that have a longer disease. We can follow the disease by weight and grip strength measures. Here you have an example of the disease according to the weight measures. So at the beginning, we have a pre-symptomatic phase where the weight is going up and the disease onset is characterized by the weight peak. And then the weight is going to go down progressively. We define the early symptoms at with the 10% of weight loss, the late symptoms with the 15% of weight loss, and the end stage of the disease is characterized by the complete paralysis of the mice. So first of all, what we did is actually looked at what's going on in the sciatic nerve of ALS mice. And you can see here staining for the macrophages. And already at the pre-symptomatic stage, we have a difference that you can see also here compared to a black 6 mice. You can see that the macrophages already change in morphology. They are becoming phagocytic. And you can see the huge increase that we have along the disease. So that shows that macrophages are actually reacting to the disease in mice. Then we wonder what's happening in human. And so we had access to ALS patient tissues thanks to Daniel Seyon, the neuropathologist associated to the team. And so we have been able to confirm that in those patients, there were an activation of microglial cells in the ventral horn of the spinal cord. But what was really interesting for us is that we did see an infiltration of activated macrophages in the ventral roots that is actually containing the motor neuron axons that were completely absent of the dorsal roots. And more interestingly, we also had an infiltration of activated macrophages in the nerve patient that is absent in controls. So both in mice and humans, it seemed that macrophages are actually reacting to the motor neuron degeneration. And what was really important for us is that in human, it's not only related to SOD1 mutation patients, but also other familial cases and also sporadic patients. So the second question that we asked was whether those macrophages are staying only at the periphery or if they are infiltrating the central nervous system during the disease, which is still a highly debated question in the field due to the different techniques that have been used. So in our project, we use a busulfan, which is a chemotherapy agent, to deplete the bone marrow of the mice and to be able to replace the cells with actin GFP cells. We use the busulfan because in our hand, in control condition, it didn't lead to any artifact infiltration in contrast to the irradiation that is often used. And so we performed this bone marrow transplantation in SOD1-G93A and SOD1-G37R mice. And we followed first in the blood. And we showed that at disease onset, in both lines, we had more than 95% of the blood monocytes that were GFP positive. So the replacement was actually really efficient. We then looked at what was going on in the tissues, and in particular, the tissues that are affected in ALS, the sciatic nerve and the muscle. And as you can see, again, both in 2 lines and in both tissues, we had, starting at this onset, approximately 70% of the macrophages that were replaced. So again, we had an efficient replacement both in the blood and in the peripheral tissues. And so we then looked at what's going on in the spinal cord. So here is a timeline of the spinal cord of the SOD1-G93A mice. So it's the shorter disease mice that where you can see that there's not a lot of infiltration. We have a slight and transient increase at the late symptomatic stage here that completely is gone at the end stage. When we looked at the longer progressing mice, the SOD1-G37R mice, we had a small increase at the early symptomatic stage that were maintained until the late symptomatic stage here, and an increase at the end stage of the disease. If you look at the graph overall, we had less than 40% of the macrophages at the most higher rate of infiltration that was actually GFP, meaning that most of the cells are still the endogenous microglial cells. And so we looked at the proliferation rate of those cells, and what you can see here is that the GFP cells that are in the spinal cords are much more proliferative than the endogenous microglial cells. So finally, the cells that you can see here are both the result of infiltration, but also the result of an actual proliferation in situ. So overall, the infiltration remains low, but it depends on disease evolution. And so we went on with the SOD1-G93A mouse model to look at what we can do and if we can modulate those peripheral macrophages at the periphery. So we used, again, this bone marrow transplantation protocol, and we did different groups. We did a group grafted with SOD1 G93A bone marrow as our negative control. The second group grafted with the actin GFP cells were actually, they didn't have any human SOD1. And the third group, we used the SOD1 wild type, which was also GFP bone marrow. So we use this SOD1 wild type because the SOD1 is, again, detoxifying enzyme, so it's rather a protective protein. And it has been shown in spinal cord lesion that the overexpression of SOD1 is actually protective. And in 2007, when it has been shown that when the SOD1 was actually downregulated in the peripheral Schwann cells, that led to an acceleration of disease progression. So we thought that maybe overexpressing the SOD1 wild type at the periphery could actually be beneficial. So we followed those mice at the different disease stages of the disease, and we looked at what's going on in the tissues. So here you have the sciatic nerve on the left and the spinal cord on the right. And the sciatic nerve, you can see that there were actually no difference in terms of macrophage reactivity for the group grafted with the GFP, actin GFP bone marrow. But we did have a reduction starting at the pre-symptomatic stage of the macrophage reactivity for the group grafted with the SOD1 wild-type GFP bone marrow. And more importantly, when we looked at the microglial cells, again, no difference has been noticed with the controlled GFP cells. But when the mice were grafted with SOD1 wild-type GFP bone marrow in blue, you can see that there was a reduction of the microglia activation starting at the early symptomatic stage through the end stage of the disease. So by replacing those mutant macrophages at the periphery by more trophic macrophages, we were able to modulate the peripheral inflammation in the nerve, but also the central inflammation in the spinal cords. So we then looked at how the disease were modulated, and so we measured the age at each mice when each mice is actually reaching the symptomatic stage. And you can see that by both the weight measurement and the grip strength measures, the most efficient effect was with the SOD1 wild-type GFP group that was delayed in reaching the symptomatic stage. However, this beneficial effect here was not enough to actually increase the survival of those mice. So we were a little bit disappointed for sure. But what was interesting is that both at the disease onset and the early symptomatic stage, when we measured the score of the grip strength, we had a higher grip strength score for this group grafted with the SOD1 wild-type GFP. So we had this beneficial effect, but we were not able to actually target the end of the disease. So what we did is that we wondered whether instead of grafting the mice at the pre-symptomatic stage, what would happen if we graft them later at the disease onset? And so what we did is basically the same thing. We used the same bone marrow transplantation protocol. We grafted at onset a group with SOD1 G93A bone marrow and the SOD1 wild-type GFP bone marrow, and we followed the mice again until the symptomatic stage and the end stage of the disease. And so again, we looked at what was going on in the spinal cord and the sciatic nerve, and in both tissue, you can see that we had a reduction at both stages of the macrophage and the microglia reactivity. So that was in line with what we previously saw with the pre-symptomatic graft. And this time we were actually able again to delay the symptomatic stage, but we were also able to increase the survival of those mice. So by replacing the macrophages, we were able to target the survival of those mice. And something that was really striking and interesting for us is that the timing that you choose to modulate your macrophages is actually really important, because by grafting later at the onset, we were actually able to have a more beneficial effect. So starting from this observation, we wanted to know how the macrophages are reacting during the disease, and because we truly believe that it's important to understand when you have to modulate the macrophages in what way or another. So what we did is we isolated the macrophages from the sciatic nerve and the microglia from the spinal cord at the different disease stages, and we performed a transcriptomic analysis. And so the first really surprising results for us is that regardless the stage of the disease, we had a huge difference between the macrophages and the microglia. They were completely reacting differently to the disease. And so when we looked at how the genes are evolving during the disease, you can see that in the macrophages, we had 18 different clusters of genes with different distinct evolution during the disease, while in the microglia, we had only 5 different clusters. So that were showing that, again, the macrophages were reacting completely differently, and the need to really understand their own reactivity towards the motor neuron degeneration. Looking at the sciatic nerve macrophages in terms of gene regulation more deeply, we were actually able to show that the changes in gene expression was actually really abrupt between the different disease stages. We had changes like between downregulation, like upregulation, and going downregulation again. And that was completely different for microglial cells because they had a more continuous reactivity going from downregulation to a gene that are going to upregulate progressively during the disease. So then we looked a little bit more into the macrophage reaction details. And so what we did is we dissected the different phases of the disease between the different stages. And interestingly, what we saw is that at the beginning, between the pre-symptomatic and onset, you can see that most of the genes that were actually regulated were linked to the transcription, transduction, protein transport, which is probably a sign that the macrophages are reacting to the motor neuron degeneration really early on. And so moving forward between the onset and the early symptomatic stage, those genes were still going on, but we had the upregulation of more genes that were linked to DNA repair, DNA damage stimulus that is probably linked to the stress response that the macrophages are facing because of the motor neuron degeneration. Interestingly, we had also genes that were related to cell cycle and cell division, which was the confirmation for us that the bigger increase in macrophage numbers is actually between the onset and the early symptomatic stage, and that's what we saw with our stainings. And finally, between the early symptomatic stage and the end stage, we had the beginning of the immune gene response with the immune system process and various GOTEMs related to the immune system, showing that this immune system is actually going, response is actually going really late during the disease and during the symptomatic phase, which was something that was really already known with microglia and macrophages. So that was really, well, we were really surprised by this, such a difference between the two populations. And so then we wonder what's actually happening when we are doing those grafts and how those different genes that we saw are modulated when we are doing this beneficial graft. So what we did is that we did a group, again, grafted with either SOD1 G93A bone marrow or SOD1 wild-type GFP bone marrow. And this time we collected the macrophages and the microglia at the early symptomatic stage and at end stage. And we performed another transcriptomic analysis. And what we saw is that in the macrophages, we had a huge downregulation of genes. Most of the genes were actually downregulated in the macrophages. That was really striking. And compared to the SOD1 G93A mice, you can see that like most of the pathway were completely downregulated. Those pathways were actually including the TGF-beta signaling, IL-6 signaling, integrin signaling, so a lot of inflammatory pathways that were completely downregulated in the macrophages. And then we looked at what was going on in the microglia. Again, when we looked at the pathways that were the top 25 pathways that were regulated, we had a downregulation of many inflammatory pathways, neuroinflammation, the TREM1 signaling, the pathway related to T lymphocytes biology. But the most surprising thing for us is that we had a huge upregulation of the synaptogenesis signaling pathway and the oxidative phosphorylation. And this pathway was really interesting for us. And so we looked a little bit more into details into the genes, and we had more than 1,000 genes that were completely going from downregulation in the SOD1 gene93A mice to upregulation in the grafted mice. And among these genes, we had more than 100 genes that were related to the synaptic pathways, and 39 genes that were belonging to this synaptogenesis signaling pathway here that are linked here. With those figures, we can clearly see that the microglia, when we change the macrophages and boost the macrophage at the periphery, even the microglia were completely changing the way they are behaving during the disease, and they were shifting towards a more neuroprotective phenotype. So as a conclusion, we have shown that the peripheral macrophages get activated during ALS disease in mouse models, and they are also reacting strongly in the tissues of ALS patients, and not only the SOD1 patients, but also other patients with other mutation or sporadic patients. We have shown that the infiltration is low, even though it depends on disease duration. And so we have been able to boost the protective phenotype of the macrophages to improve ALS physiopathological signs and improve mouse survival. The last really surprising results was that we had a huge difference in terms of reactivity between the microglia and the macrophages during the disease course. And by replacing those mutant macrophages by more trophic macrophages, we were able to change this reaction towards a less inflammatory profile and a more neuroprotective profile for the macrophages. On that, I would like to thank the team. This project has not been done by myself alone. Also our collaborators and the fundings agencies. And I thank you for your attention.
Great. Thanks a lot, Aude. And also congratulations to you for such a tremendous amount of work into your team. It's quite a feat. So we can take some questions in the chat box, but I guess I can kick it off. You touched on this point briefly in the discussion session of your paper, but I'm wondering, it's actually quite surprising that the onset grafting was more beneficial than the pre-symptomatic grafting. Do you think that's just because the positive effects of the grafted macrophages sort of fade out over time and don't quite make it into later stages of disease progression?
So that was actually my my hypothesis, and that's why we went to test what's going on when we are doing that later. I think there's two things. The first thing that is that ALS is the chronic disease, and we are not treating ALS by doing that. So of course, the macrophages are still in the neurotoxic environment, and so I'm assuming that if the mice were going really, I mean, the mice are still dying. So they are still in this environment, and I assume that at some point it's not sufficient to actually prolong for a very long time. So what we are trying to do now in the lab is to find other targets that we can modulate even more efficiently. The second thing is, I think, which I think is really important, is that when we try to modulate microglia, macrophages, we tend to do that, especially in disease, we tend to do that since at the beginning. And what I think is really important is that they have, you see that the macrophages, they have way different genes evolution. And I think it's really important to know how the genes and how your target is evolving during the disease, because it can be protected at some point, and because, I don't know, there's a huge upregulation, it can become toxic. And so you need to quite try to understand when you have to target your target to have the most beneficial effects, I think.
Yeah, for sure. That seems to be true of a lot of neurodegenerative conditions. So there was a question, I think it was from Jerry. He said he might have missed this, but what was the sex of the mice that you used in the study?
We used female SLE1.
On a technical question. How did you identify and isolate sciatic nerve macrophages?
We use the Miltony CD11B beads. So it's basically like a microglia isolation. We just triturate the tissues. We didn't use enzyme because we didn't want to overactivate the cells. And so just beads and you get the cells. And, yeah.
I'm actually wondering, how do you think that the change in peripheral macrophages are influencing microglia in the CNS? So do you think it's just like a change in circulating factors and overall inflammatory state in the periphery that is influencing the CNS? Or do you think perhaps that there are some neuron-derived cues that are influencing sort of like as a conduit between what's happening in the periphery and what's happening in the CNS that are engaging microglia in a different way and promoting a different transcriptome?
So you basically answered the question with my hypothesis. I have two hypotheses for that. We didn't test it, but the first hypothesis that I have is that basically because the macrophages are more protective, the motor neuron is basically better, and so the dialogue that he has with the microglial cells in the spinal cord is different, and so there's a like positive loop where the microglia become a little bit more neuroprotective because the motor neuron is a little bit better. That's my first hypothesis. The second is that what was interesting in our transcriptomic results, we had a huge amount of genes related to exosome that were upregulated. And so that's something that I would be really interesting to look at, is that how if those exosomes can participate in like sending a message from the periphery to the central nervous system saying, hey, like we are better here, just try to make your work up there and try to save the motor neurons. So yeah, that's something that I think, well, I cannot know for sure, but the exosome might be really interesting to look at.
I'm sorry I took your question, Jessica. I'm very sorry, but I hope that answers it for you. And last question, I hope it's really short before we take off. I'm wondering, obviously, like you said, it's probably a lot easier to target pharmacologically macrophages than it is to try and get something into the CNS and then target microglia specifically. And with no background knowledge about ALS literature at all, so I apologize. Has it been shown whether or not modifying microglia in the CNS can also reciprocally modify macrophages in the PNS like you've shown here?
So to my knowledge, no. There are for sure microglial cells that have been targeted in different ways, but until recently, people were not that interested in what was going on in the periphery. It started to, when I basically started my PhD, the papers started to come out and look at the macrophages. So I don't have any studies in mind that looked at like if you target the microglia, does that change the macrophages? The thing that is, I think, when you target the microglia, the issue is, I mean, the issue is not an issue, but macrophages and microglia are very are still similar cells. They have a lot of common gene expression, and so I think it's hard to target only microglia and not macrophages. So I think that would be hard to answer because you might change both cells profiles.
Maybe the next Nature Neuroscience paper.
Hopefully.
All right, thank you so much for joining again, and thank
you very much.
And thanks a lot to our audience. Jerry's in posting, blasting some advertisements for the next seminars. But the next Open Box Science Thursday webinar will be April 22nd at 12 Eastern time. Thank you very much to our audience for attending and to our speakers for wonderful presentations. And thanks to our master host, Jerry, for inviting all of us. Take care, everyone.